Assalamu alaikum and Bismillah Rahman Rahim. This is Dr. Narmeen Haseeb, and today we will be discussing about the changes during muscular contraction. So let's see what are the objectives of our today's lecture. Today we will learn and discuss about the resting membrane potential, the action potential, and how does the excitation contraction coupling occurs, what is the role of troponin and tropomyosin in the action potential, and how does the sliding mechanism happens. This video will be recorded in two parts. So the first part will contain the topics resting membrane potential and action potential. So in the very start, we need to know first thing, which is that a muscle contracts and there is a long story behind it. You just think of doing something and there is a series of electrical changes which happens in the inside of your cells which allows the muscles to contract, and that is all upon the action of central nervous system, upon the action of brain. So there is a lot happening. Even if you move your hand like this or this, if you decide to do something to move ahead or or backward or forward, so all of this is just not this. A lot of the series of events are happening in your body. So today we will be only focusing on what are the events or what are the electrical changes which make the muscle move, which make the muscle contract and do things. So electrical events which occur in the muscle during resting condition as well as active conditions are the basically the series of actions. So electrical potential in the muscle during resting condition is called resting membrane potential. If your muscle is at resting state, it does not mean nothing is happening inside. A lot is happening inside, and so many things are still, so many ions are involved and electrical activity is involved, which is maintaining the resting state of the muscle. And electrical changes that occurs when you are in active condition, when you're trying to move or you're moving, those changes are known as action potential. So when the electrical potential which is responsible for the resting state is known as resting membrane potential, and the electrical changes that occur when you are active are basically called, when the muscle is stimulated, are basically called as action potential. So, electrical potentials in muscle or any living tissue are measured by using a cathode ray oscilloscope or computerized polygraph. And you just measure, you get to know how the muscle gets its electrical potential. So, first of all, we need to know what is resting membrane potential. So, resting membrane potential is defined as the electrical potential difference across the cell membrane between inside and outside of the cell under the resting conditions. In resting membrane potential, it's simple. We need to know what's happening inside the cell and what's happening outside the cell. And it is also called as membrane potential, transmembrane potential, potential transmembrane difference, and transmembrane potential gradient. So for this purpose, to measure the resting membrane potential, what we do when we take two electrodes and we connect it with a cathode ray oscilloscope, and with the help of a suitable amplifier, which is placed all over the muscle fibers, uh, and what you get to know that there is no potential difference. There is zero potential difference when you just placed it on the surface of a muscle fiber. But if you basically insert the electrodes into the interior of the muscle fibers, there will be a potential difference that will be noted, and the potential difference will be collected from the across the sarcolemma. And what you will see is negativity inside the cell and positivity outside the muscle fiber. So what we came to know over here, the potential difference inside the muscle. Inside the muscle is negative and outside the muscle is positive during resting membrane potential. And this potential difference is just constant. In resting membrane potential, there won't be any difference. Why? As long as the muscle is in the resting membrane potential, there won't be any difference. So the condition of muscle during resting membrane potential is also called as polarized state, when the potential difference is constant and when there is positivity outside and negativity inside. This is all uh, known as the polarized state. So in human skeletal muscles, the resting membrane potential is minus 90 millivolts. If we talk about neuron, if we talk about the cardiac muscles, and if we talk about the skeletal muscles, all three type of muscles have different type of resting membrane potential. So skeletal muscles got minus 90 millivolts as their resting membrane potential. Ionic basis of resting membrane potential. Now we will see how is it's negative inside and positive outside. What makes it negative inside and positive outside? Development and maintenance of resting membrane potential in a muscle fiber or a neuron are carried out by a movement of ions, which produce ionic imbalance across the cell membrane. So this is all basically because of the movement of ions. And ions move inside the cell and outside the cell. So obviously there are some mechanisms which are maintaining this ionic imbalance. And this ionic imbalance results in more positivity outside and more negativity inside. It does not mean that if it's negative inside, there is not any kind of positive charge inside. But basically, what when we talk as a whole, like what's more positive inside and what's more positive outside. So ionic balance is produced by two factors. The first factor is sodium potassium pump, and the second factor is the selective permeability of the cell membrane. These factors are very important as they are the ones which decide how the inside of the cell and how the outside of the cell is going to be. So first of all, we need to know how the sodium potassium pump works. Sodium potassium ions are actively transported in opposite directions across the cell membrane by means of an electrogenic pump called the sodium potassium pump. So, we discussed the sodium potassium pump in the very start of our course, and we know how the sodium potassium pump is present between the cell membrane and how it actively pumps the potassium and sodium ions across the cell membrane. So what does it do? The function of sodium potassium pump is to move three sodium ions out of the cell and two potassium ions into the cell. And all of this happens with the help of ATP. So ATP basically allows three sodium ions to move out of the cell and two potassium ions to move into the cell. Since more positive ions, which are cations, are pumped out than inside, a net deficit of positive ions occurs inside the cell, and all of this leads towards negativity inside and positivity outside the cell. So, what's happening? More sodium are moving out of the cell. Since sodium are positive and potassium are positive, both are positive. But when three sodium are moving outside, they're more in number, and two potassium are moving inside, they are less in number. So there is a net deficit of positive charges inside, which makes the inside of cell more negative and outside more positive. Now, how the cell membrane selectively permeates ions? Permeability of cell membrane depends largely upon the transport channels. We have also discussed about the transport channels in the very start of this course, and we know there are a number of transport channels present along the cell membrane. So, the permeability of cell membrane depends all upon the transport channels. So these transport channels are selective for the movement of some specific ions, and their permeability to these ions also varies. Most of the channels are gated channels, and some of the channels were ungated. We also studied that. So the specific ions can move across the membrane only when these gated channels are opened. So we have two types of channels which are involved: channels for major anions like proteins and leak channels. So let's discuss these channels individually. First, we have channels which are major channels for proteins, for anions, for the substances which are negatively charged, like proteins. Proteins are negatively charged. Channels for some of the negatively charged large substances, such as proteins, organic, phosphate, and sulfate compounds, are absent or closed. So mostly the channels which are for negatively charged substances are usually absent or closed. So such substances have to remain inside the cell. Obviously, when they do not have any gate to pass through the wall to pass through the cell membrane, they won't go anywhere. They have to stay inside. They cannot go from another gate, which is not supposed to be for them. So they play a major role in development and maintenance of negativity inside the cell. Obviously, when the negative substances will remain inside the cell, they will maintain the negativity inside. That's a simple phenomena. Then at second point, we have leak channels. Leak channels are the passive channels which maintain the resting membrane potential by allowing movement of positive ions, which are the sodium and potassium, across the cell membrane. Apart from sodium and potassium pump, we have leak channels. They are different from sodium and potassium pump. And they what do? They are the passive channels. So they allow the free movement of sodium and potassium ions. Anytime sodium ions and potassium ions can pass through them, can pass through across the cell membrane. So three important ions, sodium, chloride, and potassium, are unequally distributed across the cell membrane. Sodium and chloride are more outside, and potassium is more inside. Since chloride channels are mostly closed in resting conditions, so chloride are retained outside the cell. Thus, only the positive ions, sodium and potassium, can move across the cell. So at this moment, we are talking about the leak channels, which allow free movement of sodium, potassium, and chloride. About sodium and chloride, they usually remain outside, and the potassium usually remain inside. And when we specifically talk about the resting conditions, so chloride channels are retained outside because they are mostly closed. Sodium is actively transported against the concentration gradient out of the cell, and potassium is actively transported into the cell. So the very important thing that we know is sodium is actively transported outside the cell and potassium is actively transported inside the cell. However, because of the concentration gradient, sodium diffuses back into the cell through the sodium leak channels and potassium diffuses out of the leak cell through potassium leak channels. So no matter who is going outside and what is coming inside, at the end of the day, because of the leak channels, both will come back to its place. So here we have this diagram, which you can see. This is the cell membrane, which have the sodium potassium pump. And this is intracellular surface, and this is extracellular surface. And all the potassium ions inside are moving outside, and all the sodium ions has to move inside. In resting conditions, almost all the potassium leak channels are open, but most of the sodium leak channels are closed. Since right now we are talking about resting membrane potential, so in resting conditions, uh, about leak channels, the potassium leak channels are always open, but sodium leak channels are usually closed. Because of this phenomena, the potassium which are transported actively into the cell can diffuse back out of the cell in an attempt to maintain the concentration equilibrium. But obviously, when the leak channels for sodium are closed, they cannot be transported out of the cell. Only a small amount of can diffuse back into the cell. This means in resting conditions, passive potassium efflux is much more greater than the passive sodium efflux and the influx. And the reason is that sodium leak channels are usually closed and potassium leak channels are open most of the times. So it helps in establishing and maintaining the resting membrane potential very well. After the establishment of resting membrane potential, inside negativity and outside positivity, the efflux of potassium stops in spite of concentration gradient. So all of this is because of two reasons. Positivity outside the cell repels positive and prevents further efflux of ions. Negativity inside the cell attracts positive and prevents further leakage of these ions outside. So it's all very obvious that the charges are going to repel each other and prevent the further efflux or influx of these ions. Now, what is the importance of intracellular potassium ions? The concentration of potassium ions inside the cell is about 140 milliequivalents per liter. So it is almost equal to that of the sodium outside. So we know one thing right now, that is the potassium which is outside the cell have 140 milliequivalent per liters concentration. And similarly, the sodium which is inside the cell has the same amount of concentration, which is 140 milliequivalents per liter. The high concentration of potassium inside the cell is essential to check the negativity. However, normally the negativity inside the muscle fiber is minus 90 millivolts, and in a nerve fiber it is minus 70 millivolts. So this concentration is responsible in maintenance of the negativity and the concentration. Uh, and this concentration maintains the negativity inside the muscle fiber as minus 90 millivolts, and inside the nerve fiber as minus 70 millivolts. So we have importance of intracellular potassium ions, as this is because of the presence of negatively charged proteins, organic phosphates and sulfates, which cannot move out normally. So when the proteins, organic phosphates and sulfates, which cannot move out normally, because we discussed that the channels which are for anions are usually absent or closed, so they cannot move outside. Suppose if the potassium is not present or decreased, the negativity increases beyond 120 millivolt, which is called hyperpolarization. And at this stage, the development of action potential is either delayed or it does not occur. So, in order to maintain the resting membrane potential, it is very important for potassium ions to maintain the negativity inside the cell, and they always maintain the negativity inside the cell. And the channels for proteins and negatively ions which are present inside the cell are absent and closed, so they also remain inside the cell. And the potassium also remains inside the cell, which maintains the negativity as minus 90 millivolts, because if it increases more than 120 millivolts, it will lead to hyperpolarization, which in turn will not allow the muscle to contract normally. So that was all about resting membrane potential. We discussed that during resting membrane potential, sodium and potassium ions are responsible in maintaining the negativity inside and positivity outside, which keeps the muscle in a resting state. Now we will discuss how the muscle contracts and how it relaxes, and all of this will be found in action potential. So, as we discussed, what is action potential? Action potential is basically a series of electrical changes that occur in membrane potential when the muscle or nerve fiber is stimulated. So, whenever it is stimulated, action potential occurs in two phases: depolarization and repolarization. Depolarization is the part when the muscle contracts, and repolarization is the part when the muscle relaxes. So, the muscle contracts during depolarization and the muscle relaxes during repolarization. So, what is depolarization? First, we will discuss the depolarization in detail. Depolarization is the initial phase of action potential in which the inside of the muscle becomes positive and outside becomes negative. So, what was previously in the resting membrane potential will be changed, and um, be it will get opposite. In resting membrane potential, the inside was negative and outside was positive. Now, in depolarization, opposite things are happening. The inside becomes positive and outside becomes negative. So the polarized state of the muscle is abolished, it's finished, and this results in depolarization. So you can see this. This is minus 70 millivolts, the resting membrane potential. When the stimulus is given to the muscle, here it depolarizes the muscle, thus changing the electrical state of the cell. Now, what is repolarization? Repolarization is the phase of action potential in which the muscle reverses back to the resting membrane potential. That is, within a short time after depolarization, the inside of muscle becomes more negative and outside becomes more positive. So the polarized state of the muscle fiber is re-established, and the re-establishment leads towards the resting membrane potential again. So and during repolarization, inside becomes again negative and outside becomes again positive. And this is important because once the muscle has contracted, we have to bring it down to the condition when the muscle was when the muscle can go in the resting membrane potential. So you can just see it over here. This is repolarization. Now we will discuss about the action potential curve. What is action potential curve? The action potential curve is the curve which shows the graphical registration of the electrical activity that occurs in an excitable tissue after the stimulation. It shows three major parts: the latent period, the depolarization period, and the repolarization period. So the resting membrane potential in skeletal muscle is minus 90 millivolts, and it is recorded as a straight baseline. First of all, we need to have a look on this curve. This curve is known as action potential curve of a skeletal muscle. What you see over here, this is minus 90 millivolts. And from here, this is stimulus artifact, which we will be discussing. And then this is firing level. Then this is depolarization. Over here is overshoot. This is spike potential. Now the muscle is relaxing. This is after depolarization. This is after hyperpolarization. So we will be discussing all these terms one by one. So first of all, what is latent period? Latent period is the period when no change occurs in the electrical potential immediately after applying the stimulus. It is a very short period with duration of 0.5 to 1 millisecond. And that's what you're just seeing. This one is latent period. Once you apply the stimulus, but the muscle takes some time to basically show some response, and that period is latent period. Then we have stimulus artifact. When a stimulus is applied, there is a slight irregular deflection of baseline for a very short period. This is called as stimulus artifact. The artifact occurs because of the disturbance in muscle due to leakage of current from stimulating electrode to the recording electrode. The stimulus artifact is followed by a latent period. So initially, before you when you apply the stimulus, here you're applying the stimulus, and before the latent period, the muscle shows a little bit of deflection, and that is just because of the change in the electrical activity because of the leakage of the current um, from the recording electrode to the muscle. Then we have depolarization. The depolarization starts right after the latent period. And initially it is very slow, and the muscle is depolarized for about 15 millivolts. And you can just see it over here. From here, the depolarization starts, but this one is very slow. It moves very slowly for almost about 15 millivolts. From minus 90 to minus 75 almost, it has moved minus 15 millivolts, and very slowly. After the initial slow depolarization for minus 15 millivolts, that is up to minus 75 millivolts, the rate of depolarization increases suddenly. The point at which the depolarization increases suddenly is known as firing level. So this was the slow depolarization for almost about minus 15 millivolts. And then what you see over here, this is the slow this is the point where the muscle shoots up, and it fires up, and it depolarizes quickly, suddenly, and very rushingly. So this point is basically the firing level. From firing level, the curve reaches isoelectric potential, which is the point of zero potential, rapidly, and then shoots up, which is basically it overshoots up beyond the potential, beyond the level of zero potential, up to plus 55 millivolts, which is called as overshoot. So you see the muscle comes from negativity, and it shoots up. It does not stop here at zero potential. It goes up and up almost at the level of plus 55 millivolts. So this shooting up beyond the level of potential, zero potential, is known as overshoot. So now what is repolarization? When depolarization is completed at plus 55 millivolts, the deep repolarization starts. And initially, the repolarization occurs rapidly and then it becomes slow. So, what is spike potential? Spike potential is the rapid rise in depolarization and rapid fall in repolarization, and together this is known as spike potential. And this phase lasts for zero point 0.4 millisecond. So you can appreciate this in this curve that this rapid rise and rapid fall over here altogether of depolarization and repolarization is known as spike potential. And you see after the plus 55 over after the overshoot of plus 55 millivolts, the depolarization uh, quickly turns into repolarization. Now we have after depolarization or negative after potential. Rapid fall in repolarization is followed by slow repolarization. It is called after depolarization or negative after potential. So what you see over here is that after depolarization, after the quick repolarization, it's not stopping here, and it's slowly still in the repolarization state. So the slow repolarization state is known as after depolarization. After hyperpolarization or positive after potential. So when the muscle again reaches to the resting level of minus 90 millivolts, it becomes more negative beyond the resting level. And this is called as hyperpolarization or positive after potential. This lasts for almost more than 50 milliseconds. After this, the normal resting potential state is restored slowly. So what you're seeing over here, that after the slow repolarization, the muscle, which is at minus 90 millivolts, the muscle is still slowly relaxing, still in the phase of repolarization, and it goes into hyper repolarization. So, and after hyper repolarization, it again goes back into the state of resting potential. So that's all what and how the muscle shows its action potential curve. It starts right there from the minus 90 millivolts to the after the stimulus artifact, it shows latent period, then it slowly depolarizes for minus 15 millivolts, and right at the firing level, it shoots up with depolarization, and beyond the potential difference level, zero potential level, it overshoots, then it's this is a spike potential, and then it shoots down, which is repolarization. It goes for repolarization further, which is after depolarization, and for further repolarization beyond the level of minus 90 millivolts, this is after hyperpolarization. And after the level of after hyperpolarization, the curve again goes in the state of resting membrane potential. So now we will discuss what are the ionic basis of action potential. Basically, the voltage-gated sodium channels and the voltage-gated potassium channels play an important role in development of action potential. So during the onset of depolarization, the voltage-gated sodium channels open and there is slow influx of sodium ions. When the depolarization reaches 7 to 10 millivolts, the voltage-gated sodium channels starts opening at a faster rate. It is called as sodium channel activation. When the firing level is reached, the influx of sodium channel is very great and it leads to overshoot. But the sodium transport is short lived. It is because of the rapid inactivation of sodium channels. Thus, the sodium channels open and closes quickly. And at the same time, the potassium channels start opening, and this leads to efflux of potassium out of the cell, which is called repolarization. Unlike the sodium channels, the potassium channels remain open for a longer duration. These channels remain open for a few milliseconds after the completion of repolarization. It causes efflux of more number of potassium ions, producing more negativity inside, and it is the cause for hyperpolarization. So, in short, which type of ions are involved and at what time they are involved? It's just simple. Sodium ions are responsible for depolarization, and potassium ions are responsible for the repolarization. And it's always like that. influx of potassium ions is responsible for depolarization, and efflux of potassium ions are responsible for repolarization. So now I will show you a video showing the action potential of the skeletal muscle.
Membrane potential, or membrane voltage, refers to the difference of electric charges across a cell membrane. Most cells have a negative transmembrane potential. Because membrane potential is defined relative to the exterior of the cell, the negative sign means the cell has more negative charges on the inside. There are two basic rules governing the movement of ions. They move from higher to lower concentration, just like any other molecules. Being charge-bearing particles, ions also move away from like charges and toward opposite charges. In the case of the cell membrane, there is a third factor that controls ion movement: the permeability of the membrane to different ions. Permeability is achieved by opening or closing passageways for specific ions, called ion channels. Permeability can change when the cell adopts a different physiological state. Consider this example. Two solutions of different concentrations of sodium chloride are separated by a membrane. If the membrane is equally permeable to both sodium and chloride, both ions will diffuse from higher to lower concentration, and the two solutions will eventually have the same concentration. Note that the electric charges remain the same on both sides and membrane potential is zero. Now let's assume that the membrane is permeable only to the positively charged sodium ions, letting them flow down the concentration gradient while blocking the negatively charged chloride ions from crossing to the other side. This would result in one solution becoming increasingly.
Action potential generation in skeletal muscle cells. The sarcolemma is the plasma membrane of the skeletal muscle. It separates the extracellular space from the intracellular space. Sodium ions are highly concentrated in the extracellular space outside of the muscle cell, whereas potassium ions are highly concentrated inside the cell in the intracellular space. This concentration of ions is what is seen at rest. Embedded in the sarcolemma are voltage-gated sodium channels, potassium-sodium ATPase pumps, and voltage-gated potassium channels. In order to initiate an action potential along the sarcolemma, a stimulus from a neuron at the neuromuscular junction causes a wave of positive charge to reach voltage-gated sodium channels on the sarcolemma. The increase in positive charge causes the voltage-gated sodium channels to open. Positively charged sodium ions flood into the cell, following their concentration gradient. This causes the inside of the membrane near to the open sodium channel to become more positively charged. This process is the depolarization of the sarcolemma. The buildup of positive charge on the inside of the membrane eventually causes the voltage-gated sodium channels to close. At the same time, voltage-gated potassium channels open, since they open only when the inside of the membrane is at its maximum positive charge. Positively charged potassium ions flow rapidly out of the muscle cell following their concentration gradient. This decreases the level of positive charge inside the sarcolemma. This return of the membrane to resting levels of charge is called repolarization. Once the sarcolemma has been fully repolarized, the potassium channels close. A sodium-potassium pump uses ATP energy to restore the concentration gradients of sodium and potassium to their normal levels.
So that was all about our today's video, in which we studied about the action potential and the resting membrane potential. The rest of the things will be discussed in part 2. So until then, take care of yourselves. Allah Hafiz.